guys, here's the HKU iGEM team.
Hello everyone, we're HKU iGEM. So this year we're working on a plastics degradation problem. So plastics, as you all know, is a global problem, right? So there are 380 million tons of plastics produced yearly, which translates to the weight of pretty much the entire human population. And the problem is that plastics degrade quite slowly, right? So for a plastic bag, it takes 20 years. For a straw, it takes 200 years. For a plastic bottle, it takes 450 years. And for a toothbrush, it takes 500 years. And this really translates to overfilled landfills, not just in Hong Kong, but globally as well. And for plastics that don't end up in landfills, it translates into plastic pollution. So here are just like three of some of the major issues related to plastic. Plastic pollution, as mentioned, the environment, human health, and tourism and economies. So let me first briefly talk about the environmental problem. So let's talk about the best case scenario, right? Where a plastic bag actually goes to landfill. The problem with that is that if you leave plastic bags out, or any plastic out, for under the sun for a long period of time, photodegradation happens, but it releases toxins into the soil, right? Such as bisphenol A, DDT, and PHS, which all have impacts to human health, as I will later talk about. And even if plastic goes through the landfill and it doesn't go through the photodegradation process, it might be combusted. So it might be burnt through incinerators, which again releases toxic substances into the atmosphere. And obviously the worst case scenario would be that plastics don't end up at landfills where they're supposed to be, and instead they end up everywhere else, right? So animals, marine creatures, birds, fishes, they can ingest plastic or become tangled in plastic or suffocate, which leads to starvation and drowning because they think that they're full, so they don't take in more food from the plastic, and instead they starve to death. So turtles, as I'm sure people have talked about because of straws and turtles and things like that, turtles, they ingest, for every one plastic item that they ingest, there's been a recent study that suggests that they have a 22% chance of dying. And as we all know, a lot of the turtles are endangered, and that's not a great outlook for the turtles. And we see that a lot of the fishes, actually 2/3 of fish species, have actually also ingested plastic. So let's talk about another best case scenario, right? So what if there is a proper disposal and management? That's very tricky actually, because then you have to consider the wind and the waves and things like that. So even if plastic is broken down properly, if it's managed properly, all the toxins are managed, you have to consider wind, right? Where microplastics, which are plastics smaller than 5 millimeters, they get transported back into the natural environment. Another alternate best case scenario might be recycling, but the problem with that is that sometimes, a lot of the times actually, toxins are also released in the process, and actually very few of the original plastics are actually used. There's also a climate impact where a lot of greenhouse gases are emitted as part of the recycling process. So it's not contributing very well. It's not doing very well when it comes to climate change impacts either. So this is just a summary of all the things that I've just mentioned. And you see that even in the best case scenario, in most scenarios, there's going to be toxins released. And in most scenarios, it's going to go to the natural environment at some stage anyway. So now let's talk about human health. Right, so I previously talked about some of the toxins, and this is just a handy table that tells you some of the human health impacts that some of these toxins may have on humans. And we note that some populations are disproportionately more affected than others because of environmental justice issues, where minority groups tend to live closer to landfills, and places where waste is discarded. So as previously mentioned, 2/3 of fish species have now found to be, have found to ingest plastic. And we see that microplastics and toxins, they cycle through the entire ecosystem. So it goes from the soil to the water to the atmosphere to different species, and we're actually just eating all these species because we are part of the ecosystem, right? So what this means is that we are also ingesting plastic. So this goes back to the human health problems here. And finally, let me briefly talk about tourism and economies. So obviously plastics is an aesthetic problem as well, because given the choice between the image on the left and the image on the right, you would probably want to go to the place without the plastic. You just don't want to see it. And that's obviously, well, perhaps with COVID, it's not as much of an impact, but it's going to have impact on different locations as well. And this is also a global justice problem because obviously countries all over the world, they produce plastic waste and they have to dispose of this waste at some point. And a lot of the times, all this waste, it gets exported to places that are less fortunate. And China actually recently, in recent years, they put a ban on receiving plastic waste from other countries. Just because there was so much waste and they didn't know how to deal with it. So now we're shipping our waste over to Vietnam and Thailand and countries in South Asia instead. So here's the catch, right? We probably didn't need to tell you all of that. We probably all know the effects because, it's a very popular search term. The Google searches for plastic since 2014 have remained relatively stable. And this table, it shows, so 100 is the index. That shows the most popular searches, right? And you see that over time, the word plastic, it has remained at approximately 50 to 100, right? So plastic is it's pretty well known. The actual problem is that organisms are struggling to break down plastics. So evolution takes time, as we all know. And the first plastics, they emerged in the 1950s. And this means that organisms have had relatively short period of time to learn how to break down plastics. The second problem is that plastics are great, right? They're low cost, they're light, they're durable, which means that you can produce a very thin sheet of plastic and you can still carry it around. So that relates to how it's very light. It's very hygienic. There are a lot of food-safe plastics out there. They're very versatile, flexible, and there are not a lot of alternatives. And COVID is just exacerbating the issue. So there are two ways to tackle the problem. One is to decrease the demand, and the other one is to find a better solution to handle the end of life of plastics. So in terms of demand, right, these mostly relate to financial incentives, and Hong Kong is clearly not doing a great job because these plastics are sold relatively cheaply. And yeah, so as you can see that these are very questionable effectiveness in terms of plastic usage. So you can see that plastic usage remains high despite some of these financial incentives being rolled out. So the second solution would be to handle the end of life of plastics. But there are low recycling rates relative to other countries, and people don't seem to care enough in the first place. So this brings us to our project.
This year, our team aims to produce a solar-driven, self-sustaining bioreactor to digest polyethylene terephthalate, or PET for short. We're focusing on PET as it is one of the most common types of plastic, with over 17 million tons of PET used in textiles and packaging each year. That is 18 million elephants' mass worth. Utilizing the potential symbiotic relationship, mutualism, between the bacteria E. coli and the cyanobacteria Cynococcus elongatus, we hope to synthetically develop a co-culture system between the two organisms that can, number one, drive PET-associated enzyme production for plastic degradation in E. coli, and two, utilize the sucrose produced by the photosynthetic algae for E. coli as an energy source. Ultimately, the PET plastic will be degraded into monomers and taken for further processing or manufacturing into new bioplastics. Here, we show you our plan to achieve this. E. coli is frequently used in synthetic biology laboratories due to their high efficiency in expressing foreign genes. Here, we use the BL21DE3 strain as the biological machinery because of its excellent ability for recombinant protein expression. We will engineer genes that code for PETase, MHETase, as well as CSC-B, A, and K, which we will further explain in a little bit. To pair it with its photosynthetic cyanobacteria, S. elongatus, the strain we're using is PCC7942. This organism will provide sucrose as the energy source for E. coli to carry out its functions. We will also engineer a CSC-B and LAC-I gene into the organism. Firstly, we designed a plasmid that codes for PETase and MHETase. That is transformed into the E. coli. While PETase would digest PET into monomeric MHET plus some trace BHET, MHETase comes into play to further digest the MHET into tetrafallic acid and ethylene glycol, which can be utilized and resynthesized to produce new bioplastics. Only a few E. coli strains can readily uptake sucrose as an energy source, for instance, E. coli W. Most of them can only take glucose. However, we can always engineer E. coli to carry out new functions. Here, we incorporate sucrose utilizing genes into BL21. CSC-B acts as a sucrose permease that allows sucrose to be channeled in and out of the cell. And both CSC-A and CSC-K are genes responsible for the metabolism of sucrose. We incorporate these genes into the bacterial genome instead of merely transforming them as a plasmid. And we achieve this by using the lambda-red system derived from a bacteriophage. This ensures that the sucrose utilizing function would not be easily lost through the generations as the organisms propagate and carry out cell division. After we establish a stable line of sucrose-eating cells, we can then transform our plastic plasmid into these cells to reach maximum efficiency. Of course, we will also want to optimize the sucrose secretion by the cyanobacteria, since it serves as an important energy source for E. coli. CSC-B is again transformed into cyanobacteria as it creates channels and allows more sucrose to be transported out of the cells into the surrounding waters. We add a LAC-I regulatory gene as well, making this an inducible system that will be turned on by IPTG, a molecular mimic of allolactose. This ensures that the native cellular mechanism of the cyanobacteria will not be exhausted by extreme sucrose production, and it will also stop extreme production of a certain chemical that could ultimately lead to an adverse or unwanted imbalance in the system. To achieve symbiosis, our future model would include a function for E. coli to convert atmospheric CO2 into bicarbonate. Not only would this transform E. coli into a carbon sink, thus cancelling out the effects of its own respiratory functions, it also provides a readily usable bicarbonate for S. elongatus as an extra carbon source. This optimizes sucrose production and thus E. coli function, achieving a true self-sustaining and materials recycling system. According to a paper published by Nature in 2019, a team in Israel has successfully trained a strain of E. coli to survive solely on CO2 through directed evolution. However, this is only achievable when the CO2 levels are 250 times of that on Earth. And when provided with glucose, the E. coli always prefers the sugar to CO2. While the system is currently not energetically economical in an oxygen-rich environment, we can nonetheless slightly increase the carbon storing ability of E. coli and allow it to convert atmospheric CO2 into bicarbonate for cyanobacteria to use for photosynthesis. After digestion by PETase and MHETase, to recycle the monomers, we can utilize the Pseudomonas genus and their ability to manufacture bioplastics. Pseudomonas strains produce polyhydroxyalkanoids, or as you know, PHAs, as their source of energy. PHA can then be used instead of a traditionally commercially produced plastic, as this is an environmentally friendly alternative. With this, we can create not only a biological recycling system between E. coli and S. elongatus, but also a material recycling plant that saves toxic monomers from plastics and turns them into a new product. When wet lab collects enough data, the dry lab will be responsible for modeling. We take our inspiration from a paper published just this year on the co-culturing system. While this is a paper on isoprene production, the team has successfully achieved a 1-to-1 ratio of E. coli and S. elongatus in a staple co-culture medium. And this is the container that it will be held in. I know it looks like a washing machine. The co-culture will take place in the bioreactor, which includes a pulverizer for plastic, a sieve for plastic bits, and sinks with semi-permeable membranes for the co-culture system. We can easily remove the components of the reactor and clean or replace it. And this reactor will be able to operate in freshwater plants, where it will collect PET to alleviate the problem of plastics being washed out to the oceans.
Alright, so briefly on education and communication, since I know that we're running out of time, I will breeze through these slides. So we've been trying to promote STEM education for the most part, and for the most part also promote synthetic biology in our activities thus far. So we've participated in JSSE, we've had a booth there. We briefly taught a bit, we had a workshop about synthetic biology and Python programming to Rwandan children in ASYV. In collaboration with a few other iGEM teams. We are currently hosting a bioinformatics course, which covers a bit of synthetic biology as well, under the partnership with Coding Kingdom. And we are, in light of, well, from our research and from our conversations with professors and other people as well, we know that there's very limited synthetic biology policy in Hong Kong. And even at our own university, HKU, there doesn't seem to be any guidelines on how synthetic biology should be performed. So in light of that, we are also hosting a policy case competition where we hope students can learn more about synthetic biology, policy, biotech business, and other related topics and their applications. And finally, we're also hosting a cyanobacteria symposium because we are doing work related to cyanobacteria, and we hope to conduct a knowledge exchange there. So similar to UCCKE, we are going to need some help. So if you could all scan this QR code, that would really help us for our human practices, because we're trying to understand how plastic, what people think about plastic waste, and a little bit about synthetic biology attitudes as well. Okay, so same QR code here, don't worry. We were very desperate. But yeah, thank you for listening. If there are any questions, we'd be happy to answer.
Guys, I had a question. So you said you're going to be incorporating the genes for plastic degradation into the genome of the bacteria, right? But the plasmid copy number, I mean, naturally it's genomics, so it's definitely not going to have a plasmid copy number.
The plastic is a plasmid. The one we're incorporating into the genome is a sucrose utilizing genes.
Okay, so is that good enough? Like, can it withstand the sucrose structure based on that?
So we're hoping, instead of giving it two plasmids, that would certainly exhaust it.
Yeah.
So we're just hoping that within the genome itself, we can achieve a stable line first, and then introduce a plasmid.
Actually, that makes sense. Yeah. Okay. Thank you.
And how do you screen for the stable lines?
To see if it can survive on sucrose.
So you use a sucrose deficient
I think we have a sucrose kit for that.
Speaking of the sucrose, I don't quite get the idea of why you want to increase, like allow the E. coli to use sucrose as energy source, because if you can simply culture it with any other
So we're trying to get it to be a self-sustaining system. So if we have to provide it with glucose, then that's an extra input we have to do. But if we're putting cyanobacteria there, we can just use sunlight as the only energy. And if it's in the water plant, just flowing in the water, it can just receive sunlight. Cyanobacteria can produce sucrose, and the E. coli can use the sucrose for the enzyme production. Is that convincing?
But then, I'm just thinking because the cyanobacteria itself has to utilize some part of the sugars, right?
Yes.
So let's say it takes up half of the energy to sustain its own protein. And the amount of energy that it can pass out to the utilized protein. I don't know what is the number that we're looking at, but it might be quite little. And I'm not sure if that is enough to drive the survival of the
We're trying to, first we're adding more, like with the CSCP gene, we're adding more sucrose channels in the cyanobacteria. So if it produces anything in excess, it can always come out. And we also have a lac-I gene in the cyanobacteria, so it's like inducible. So it will not like just keep producing sucrose constantly, and that would exhaust the mechanisms.
In that case, would it still be self-sustainable?
In what way?
If you have to add an inducer.
I think the inducer is just for the initial stage of testing.
Oh, okay.
Just yeah.
I have a slightly, I just have an idea. Does the cyanobacteria itself produce glucose then?
Glucose? Not. I don't think so. No. Like it's not its main production.
Because what I have in mind just now is that what if you do it the other way around? Instead of allowing the E. coli to take up glucose, you produce glucose with the cyanobacteria to feed you. Just as a fact.
Good idea, but also.
Anyways, you guys don't know way more than me.
Oh no, no, it's open discussion. Yeah. But a problem could be like environmental E. coli could also grow. Environmental E. coli, like normal native E. coli in the waters, could also grow, and that could be a problem.
How big is that washing machine thingy you were talking about? I'm just gonna call it washing machine now.
It's roughly this.
Was that something you created, or is that a bioreactor?
Okay, so sorry, can you say that a bit louder?
Oh, was this something you guys created, or is it like a thing?
Oh yeah, no, this is something that we generated in SolidWorks.
Oh, okay.
Yeah.
So have you made it yet?
We've not actually made the actual prototype, but we've like 3D printed models to see if it will indeed fit properly. So this is just like the initial testing stages, but it is a prototype.
So in this tank, you're gonna put that co-culture of E. coli and cyno.
Yeah, if you can see the cylindrical drum at the door at like the bottom there, that's where we're gonna feed our co-culture, and the rest is just like the external shelling to like kind of protect it.
Where will the sunlight come from now?
Okay, so it's not shown here, but it's meant to be transparent.
Okay.
Yeah. Technical issue, sorry.
No, don't worry. So how do you 3D print that thing?
Yeah, through a 3D printer. Like, oh, you mean the actual thing?
Yeah, if you want it to be transparent, how do you? Can it be
Acrylic. We use acrylic filaments.
Okay.
Any more questions?
down the plastic, you are going to recycle the two monomers, I think it's TPA and cycle.
Yes.
Are you going to recycle them or what are you going to do?
We're adding this. Okay, so this is for the future because we're now trying to break down the plastic first because the monomers are not they're less toxic than the plastic itself. But afterwards, we want to try our best to make this a whole actual system, right? So this Pseudomonas genus is actually a really big group, but it can store PHA as a form of energy itself. So it can synthesize bioplastic from all these monomers. So if we can use that, then that's an actual cycle.
Because from what ethylene glycol is toxic, I think. So it wouldn't be very nice to have a bunch of ethylene glycol just floating out in the system.
Yes, we're hoping the bioreactor would contain everything.
So if there's no other question, then thank you very much, HKU iGEM team. Thank you. So that concludes all of our presentations, and I would like to say thank you again all so much for each and every single one of you. I understand that you guys put a lot of effort into these presentations and into your projects, and it was actually really interesting to actually get to know all of your projects better. Now, before our photo